Okay, plant breeding. Super fun. Basically involves tweezers, a microphone, uh, some kind of usually. I I always used a lens, but a lot of people don't. I apparently just had terrible eyesight. This is a very tedious task. It needs to be done in a clean environment, and you're basically taking the anthers and the pollen off, or you're removing the ovule, or you're covering the ovule. You're doing this usually in a greenhouse. Sometimes you're doing it in the field. Like I said, I have crawled along rows of soybeans and done this with tweezers and Q-tips. It's not super fun, but it is a really interesting thing that you are doing. So again, breeding, usually you get an F1. So let's say you have this interesting trait in corn, but it's everything else about the corn plant isn't great, right? Let's say it's an interesting, we'll go with sweet corn. It's extra sweet, but the plant isn't very sturdy, like the stalk is small and the ear doesn't. So you're like, well, I don't like anything else about this plant except for that the kernels are really sweet. So you're going to find that you're going to take this plant with the sweet corn and you're going to cross it with a parent that you know has a good stalk, good roots, disease resistance. You're going to cross it. Okay, we still have kind of a weak stalk, but we've got this good gene. Then you're going to cross it again with a parent. Then you're going to cross it again with a parent. Then you're going to cross it again. And finally, you're going to get all the other good things about that plant and the extra sweet kernels. Does that make sense? That is what hybrid corn looks like. You're just going to back cross it again and again and again with this parent that you like everything else about it, except it's not as sweet as it should be. So you just keep back crossing it to that parent. They're plants. They don't care about, mating with their parents. So just don't think about humans. Think about plants. Again, you're going to cross-pollinate. You're going to self-pollinate. You're going to back cross it over and over again until the F2 generation here is exactly the way you want it. So that you have a good characteristic of everything else, and you're also expressing the one characteristic that you're looking for, like in this case, the extra sweet corn. Single gene traits are easier to breed for. If we're just looking for an extra sweet color kernel, like a white kernel that is extra sweet, that's pretty easy to do. Sometimes when you do this over and over, you reduce the vigor, so it's not as the stalk isn't as thick, or it's not as disease resistant. You do risk that a little bit. But if it's not the way you like, you just keep back crossing until you get both of those the way you want them. Okay, hybrid seeds. Again, this is 2 inbred lines. You will cross this one, then you will cross these 2, then you will cross this, and you will get a double cross hybrid. So these are, let's say, are 4 traits that you really want. It's like a disease resistant, it's good kernels, it's good roots, it's good stalks. You're going to breed these 2 together, then the F1 of that is going to be here. You're going to breed these 2, the F1 of that is going to be here. You're going to breed these 2 together, and you're going to get a double cross hybrid. Almost everything in corn and soybeans are called hybrids. If you actually planted this, you would get a variety. That's why you can't always save your seed, because this seed is going to be all these parents. It's not going to be as perfect because we bred it that way. We took this pollen and put it on this ovary and the pollen tube. So saving your seed here is not going to get you an identical parent. So the F1s all have the same, all these inbred lines we know are going to have the same continuous genotype, but as you cross them, this is not going to have the same continuous genotype. By the F2, it has great variation. You cannot save your seed from year to year. Hybrid seeds are very popular. It's something called hybrid vigor. You only get it the first time, and if you try and save those seeds, it's just not going to work. Here it is again. This is how the hybrids are made. You have 2 inbred lines that those seeds are going to be planted here, and then this inbred line, this pollen is going to go into this, and you're going to get this hybrid double-crossed corn. And this is going to be superior than even if you planted these kernels, they're not going to be as good. Now let's start on propagation, sexual versus asexual. So again, sexual is propagation by seed, asexual is vegetative. This is all of the types, the cuttings, the grafting, the budding, the layering, the suckers, all of that. Okay, propagation terms. A cultivar is a cultivated variety. This is one that we have over time maintained into a variety. These are, you will see names of them. Different types of petunias, especially in the ornamental world, have a cultivar. Wave petunias are a type of cultivar. A line, this is going to be produced from true seed. This is like your ball seed. They're going to produce a line that always produces true from seed, because they're going to have growers who want to make sure that the seed, that the tomato plant that they plant is the same every time, because they're selling those tomato plants commercially. A clone is a cultivar that originated from a piece of a single plant propagated vegetatively. I don't know why I can't say that. So a clone, sometimes you can order clones. You can order just a propagated part of something, and they will send them to you. Sexual propagation, this is going to be seed production, seed formation, storage, dormancy, germination. You're going to want to know some stuff about seeds that you are using. So seed production, purity generally assured if the cultivar is homozygous and it's self-pollinated. This is ideally what you want. You want to get back to a homozygous self-pollinated plant. You're going to produce seeds from that, and those seeds are going to very consistently produce the same type. Sometimes the cultivar can be homozygous, but it will cross-pollinate, so you have to keep the female plant away from the undesired pollen producers. This is big greenhouses where they want very similar, they want all the tomatoes to be the same size, or they want all the flowers to be exactly the same color. They will have to make sure that they don't cross-pollinate. Seed production. So seed certification is a big deal. This is done by states and countries, and they have set standards. So there's somebody that will come in and look at the germination rates, come in and look at the seed purity, look at the traits that they have produced. Seed certification is kind of a big deal, and you can get a lot more for your seeds if they are certified. They can be certified as a breeder, as a foundation seed, as a registered seed, or as just a plain certified seed. So breeder seeds, these are small amounts produced under the control of a breeder, and they're sold with white tags. These are sort of unusual. Unless you want to breed, you're not going to get breeder seed. Foundation seeds, these come from breeder seeds. Also sold with a white tag, produces a registered seed, limited amount. These are extremely expensive. Not as expensive as breeder seeds, but they're pretty expensive. Registered seeds, these are the offspring of breeder or foundation seeds. The seed source comes certified, and it's sold with a purple tag. Then you have certified, which is what most people have. It's a known genetic identity and purity. It's available in large quantities. It's sold to farmers, and it comes with a blue tag. So there are white tags, purple tags, blue tags, and that is how close to the original breeder seed that you have. And of course, there's a cost for that genetic purity. Genetic quality of other crops is mostly regulated by the seed producer. So whoever produces that seed and how much they're willing to pay to get it certified, foundation seed, all that stuff, is how much the cost is going to be back to the grower. Okay, seed formation. Yes, you have to look at this. So this is the endosperm. This is basically going to be what the plant uses for nutrition, the outside of the seed coat, and then the cotyledon. These are things you have to examine when you get seeds in. If it's all shriveled and there's no endosperm, you're not going to get a plant is not going to have enough energy to germinate. If the seed coat is, not there or has been hurt, then you're definitely also going to have a problem. Same thing with a dicotyledon seed. You already have the true leaves in here. So with both of these, you can cut them open and look at them under a microscope, which most commercial, horticultural, and even agriculture will look at them to make sure that they have proper food storage, good seed coverings, the embryos look intact. Seed formation, again, flower part development. You've got an ovary, an ovule, and integuments, which are the seed coats. You've got this, the female part here, and you're going to have a pollen tube that grows down into here. You've got the two nuclei. This is what's going to be inside your ovary. You've got the endosperm cells. You've got the suspensor. You've got the basal cells. You've actually, this whole thing is the seed. You've got a nucleus. You've got different parts of the endosperm. Okay, all of that is going to contribute to your seed health. Seed storage is important.